So a very good morning to Puan Anis. My name is Denise and my matrix number is 23DBT 2005, and today this lab is on the preparation of inoculum in bioprocessing. So in this lab, we will actually learn how to grow and harvest E. coli. But before we start with it, we will actually do a pre-lab, which is done to review the principle and the processes of the fermentations in the pre-lab. So whatever that we've learned, like the principles, we would actually add it into the notebook so that when we are proceeding with the lab later on, we would be able to review the notes in the lab notebook. So as we move on, these are actually the phases of the cell growth, consisting of 4 phases. And in this pre-lab, we will learn about all of it. And it'll take about 10 hours for the growth, which will be from the lag phase to the death phase. So I'll add it to my notebook and move forward. So when I click on each of it, I get to learn about the lag phase, the cell being able to adapt to a new environment, and then the log phase, and then we have the stationary phase, and lastly the dead phase. Then we would know about the conditions of the cell growth. All of this would be needed for a proper cell growth, being the pH maintenance, oxygen, agitation, sterile environment, the nutrients, and the foam control. So all these will be added into the lab notebook for further references during the lab. So on this part, we will learn on the corresponding controls in the fermenter by clicking on each of the white boxes, like the temperature, pH, aeration, and foam. And then as usual, we add it into the notebook. So now we go more in depth on the growth curves, like lag phase, exponential, stationary, and death phase. So we have to add all this into our notebook. The lag phase being the initial growth phase, the exponential phase, the cells are grown in required volume and density, the stationary phase, the nutrients are being depleted, and the death phase. So now we move on into the experiment. The next part would be the procedure. We have to pick 4 steps. So the first one would be the thaw inoculum vial contents. The second one would be the transfer of inoculum vial contents to the shake flask. Third one would be to take note of the starting conditions and then place the shaker flask in the incubator. So I will view the notebook just to double check and see if I got all the steps right. So after I submit it, I've been able to get a proper curve. Moving on to the next part, which is the processing. So on this part, I'll just have to select the areas of the fermenters that I need to check, and then I will learn more about it. So they let us know on which places we should always check to make sure that it's in a good condition. questions like this would be asked: how we should cool it, the temperature of the fermenter. So I chose fill it with cooling fingers. And then the pH has dropped, so I decide to add a base. So the next part would bring me to the stationary phase. So before I pick these steps, I have to go through my lab notebook just one more time. Right below there, the steps are written. So I take a look at it first, and then I go in with the first one, which is add the chemicals to make cells overexpress a particular gene. The next step would be to note whether nutrients are being depleted, and the last one would be to harvest the cells. That is all. So it's been ready to harvest, and I have finished my lab simulation. That is all from me today. Thank you, madam.